When a pathogen enters the body for the first time, it's usually detected and ingested or phagocytosed by something like a macrophage. So here we see a macrophage, an antigen-presenting cell, that ingests, in this case, a bacteria that has particular antigens on its surface. These, I don't know if you can see, are triangles and squares. Once the antigen-presenting cell or the macrophage ingests it, it digests it, and then presents the foreign antigens on an MHC2. Now, it goes around in the lymph nodes or the tonsils or the spleen or gullet, and it shows this wanted poster to every cell that it encounters in that area. Eventually, it will find a helper T cell. It always has to be a helper T cell first. Finds a helper T cell that will recognize at least one of the antigens that it's presenting. And once this recognition takes place, the cells have a little conversation with chemicals. But the conversation basically sounds like this. The antigen presenting cell says, "I order you to activate," and the helper T cell says, "Yes, sir." Then the helper T cell is stimulated to multiply. So about 1 week later, we will have a whole bunch of helper T cells that have been activated by the antigen presenting cell. Now, some of these cells are going to be held aside. They will not enter battle. They will stay stationed where they are in case we encounter the pathogen again, and we call those memory helper T cells. The remainder of the cells that are produced go into battle. So these are called effector helper T cells because they're actually entering the battle. Now, if this pathogen enters the body a second time, we don't have to go through these steps anymore because we already have memory helper T cells that have been activated. And this is why when you encounter a pathogen a second time or a third time, the response of your specific immune system is so rapid that you usually don't have symptoms. But the first time, there is a long process of activation, and this is why you get sick the first time you encounter a pathogen.